All right, welcome back, everyone. Thanks for joining us for another deep dive. Today, we're diving into something really fascinating. It's changing how we see a pretty complex disease. We're looking at this surprising link, the one science is uncovering between our immune system and Parkinson's disease.
Yeah, it's a connection that, well, for a long time, it wasn't really front and center. We knew Parkinson's meant losing certain brain cells, right? And these weird protein clumps, Lewy bodies building up. But how they formed, why those specific cells died, that was a big puzzle.
Exactly. And what we're going to do today is basically take a whole bunch of source material, research papers, new studies, findings from key labs, and really dig through it together. We want to pull out the most important stuff for you. Think of it as us sort of navigating this complex info for you.
And our starting point, really the thing that kicked off this particular deep dive, is this pretty groundbreaking study from McGill University, from the neurolab there. It gives a totally new perspective on those Lewy bodies we just mentioned.
Okay, let's unpack that McGill study then. What was the big deal? Why is it considered such a breakthrough?
The really exciting part, and what was so tough before, is they actually managed to replicate Lewy body formation for the first time in real human neurons that they grew from stem cells. This is huge because, until now, we could mostly just study these clumps in brains after death, which really limited seeing how they actually form.
So they built like a living model to watch it happen in real time, a process we couldn't see before.
Precisely, yeah. They could literally watch these alpha-synuclein protein clusters forming inside the neurons. And by doing that, they could start to figure out the specific ingredients, the conditions needed for those Lewy bodies to actually appear.
And what were those conditions? It wasn't just having the alpha-synuclein protein hanging around.
That's the key thing. No. They found you needed two critical factors. You needed the alpha-synuclein, yes. And crucially, you needed an immune response happening inside that neuron.
Wait, hold on. An immune response inside the neuron.
It wasn't just about the protein folding wrong. The cell's own internal sort of immune defenses had to be triggered.
So the immune system wasn't just like cleaning up the mess afterwards. It was actually part of creating the clumps in their model.
That's what the study strongly suggests, yeah. And they found something else too, even more specific. These Lewy bodies only formed in the dopamine-producing neurons, the exact type of brain cell that dies off in Parkinson's. They didn't form in other types of neurons they tested.
Wow, that specificity is really something. It mirrors the actual disease. So, okay, being able to replicate this in human cells, see it live, and pin down the immune system's role. What did the researchers themselves, like Peter McPherson and Armin Bayati, say about the implications?
Well, Peter McPherson, one of the senior researchers, he basically said this finding really suggests that things triggering the immune system, stuff we run into all the time, maybe long-term stress or exposure to certain toxins, chronic inflammation, these things might be way more linked to the start of Parkinson's than we thought.
That feels like a pretty big shift. It's not just genetics or protein problems anymore, but how our bodies react to the world around us.
Exactly. And Armin Bayati, the first author on the paper, he added that their results kind of imply that potentially anyone could develop Parkinson's if they're exposed to the right mix of environmental triggers and immune responses. It suggests maybe you don't always need a strong genetic predisposition. The environment and immune system could, theoretically, push someone over that line.
Wow. So the environment and our immune reactions could be much more central for some people than we realized in actually kicking off the disease.
That's the huge implication, yeah. They also pointed out that understanding how Lewy bodies form in this deeper way, seeing them not just as simple protein blobs, but complex things built with immune help, that gives us totally new ideas for potential drug targets, ways to maybe slow down or stop them forming and hopefully slow the disease. This work, it was published in Nature Neuroscience, a top journal, so it gives real experimental weight to this whole idea of the immune system being an active player. And that McGill study is just, it's a perfect way in, because its findings really back up this much bigger wave of research, all pointing towards immune issues and inflammation being key factors in Parkinson's.
Okay, let's dive into that broader picture then. Beyond just the immune stuff happening inside the neuron, what else is going on immune-wise?
Okay, let's start with the brain's own immune cells. They're called microglia. Research, including looking at brain tissue after death, it consistently shows these microglia are highly activated in the parts of the brain hit by Parkinson's.
Activated? What does that actually mean? What are they doing differently?
Well, instead of just their normal housekeeping and support roles, activated microglia are basically on high alert. they start releasing floods of these signaling molecules called pro-inflammatory cytokines. Things like IL-1, TNFO, think of them as chemical alarm bells, shouting problem, and ramping up inflammation. They also put more of certain proteins on their surface, like HLA-DR, TLR12, like little flags saying they're ready to fight.
So they're actively making the brain environment more hostile.
Pretty much, yeah. And what's really interesting is that PE scans, using these special markers that light up activated microglia, they show this activation isn't just an end-stage thing. It's there really early on, sometimes years before the main motor symptoms show up. Even in people with conditions like REM sleep behavior disorder, which we know increases Parkinson's risk later.
And the significance of these superactive microglia, what's the downstream effect?
By pumping out those inflammatory signals and other sort of toxic stuff, they create an environment that actually speeds up the death of those vital dopamine neurons. So they're not just bystanders, they're actively adding fuel to the fire. Then, moving beyond the brain's own immune crew, research also shows immune cells from the rest of the body are getting involved. The peripheral immune system.
You mean like T cells and stuff, the ones that fight infections in our blood, they somehow get into the brain?
Exactly that, yeah. Studies showed different types of these peripheral cells. T cells, like you said, both CD4+ and CD8+ types. And monocytes, too, they actually infiltrate the brain regions where the damage is happening in Parkinson's. What's interesting is that the more aggressive ones, the CD8+ cytotoxic T cells, they seem to show up pretty early. While the CD4+ helper T cells, the ones that kind of coordinate the immune attack, they tend to increase as the disease goes on.
And what do scientists think these outside immune cells are doing once they get into the brain?
Well, they could be making things worse by just adding more inflammation to what the microglia already started. There's even some thinking that they might directly attack the neurons themselves, maybe mistaking them for something dangerous.
Okay, so you've got the brain's own immune system fired up, and then the body's immune army potentially crossing the border and joining in.
That's the complex picture that's really emerging. It's not just contained within the brain.
And alpha-synuclein itself, the protein at the heart of Parkinson's. We know it clumps up, but does it actually, talk to the immune system directly?
Oh, absolutely. It's not just passively sitting there misfolding, it's actively provoking the immune system.
How does it do that?
Well, research shows that the misfolded version of alpha-synuclein can act like a danger signal. It activates both the microglia in the brain and those peripheral immune cells that get in. It seems to do this by latching onto specific receptors on these immune cells, things like toll-like receptors or TLRs. These are part of the immune system's toolkit for spotting threats. When alpha-synuclein binds, it triggers these major internal alarm systems in the immune cells, pathways like NFB and something called the NLRP3 inflammasome.
So the protein itself is basically ringing the immune alarm bell, telling the system something's wrong.
Yes, that's a good way to put it. And this interaction even hints at something potentially autoimmune going on. Studies have found that people with Parkinson's often show stronger reactions from their T cells, specifically to little bits, fragments of the alpha-synuclein protein, specific regions like S129 or Y39.
Meaning their immune cells seem to recognize parts of this human protein almost like they're foreign invaders.
That's exactly what it suggests, yeah. A possible autoimmune reaction where the body's own immune system might be targeting neurons because of this alpha-synuclein. We also need to think about the bigger picture, genetics and environment. Because it looks like vulnerability to Parkinson's isn't just about how well your cells handle protein. Genetics and environment play a huge role, and often it seems they do it through the immune system.
How do genes connect to the immune system here?
Well, there's evidence that some gene variations known to increase Parkinson's risk think mutations in GBA or LRRK2 genes. They're also linked to problems or, let's say, dysregulation in how the immune system works. So the genetic risk for PD seems kind of tied up with immune system vulnerabilities.
And the environmental factors, we touched on that with McGill.
Right. Things like just getting older, problems with mitochondria, the cell's energy factories, gut health issues, especially an unhealthy mix of gut bacteria, what they call dysbiosis, and long-term exposure to certain toxins, like some pesticides. All these things can weaken the immune system's balance and promote inflammation throughout the body. There's even research linking past infections like flu or herpes viruses or specific pesticide exposures to changes in immune responses that might, down the road, encourage neurodegeneration. And we can't forget another key brain cell type, the astrocytes.
Ah, yes, astrocytes. Aren't they usually the supportive ones? the caretakers.
Normally, yes, but in Parkinson's, the evidence suggests they kind of switch sides. Unlike in some other brain diseases, here they seem to lose some of their protective functions and actually become pro-inflammatory. They start contributing to the problem.
So they go from helpers to part of the issue.
That seems to be the idea. This shift in astrocytes adds more fuel to the inflammatory fire, and it even looks like they might help spread the misfolded alpha-synuclein protein from cell to cell.
Okay, phew. That's a lot of moving parts. Microglia, peripheral cells, alpha-synuclein triggering things, genetics, environment, astrocytes. Let's try to tie this all back together. How do all these immune mechanisms specifically lead to forming those Lewy bodies we started with?
It really looks like a complex feedback loop, almost a vicious cycle. Inflammation, both body-wide and just in the brain, seems to make alpha-synuclein more toxic. It makes it more likely to misfold and clump up into those aggregates.
So inflammation creates this environment where the protein is more likely to go bad and stick together?
Exactly. Chronic inflammation makes the damage from alpha-synuclein worse and seems to directly help form more clumps. Plus, inflammation can actually damage the cell's own cleanup crews. Systems like the ubiquitin proteasome pathway, basically, the cell's garbage disposal for bad proteins.
The way cells normally get rid of junk proteins.
Yes. And when that system gets damaged, maybe by inflammation, maybe by problems with mitochondria, which are also linked to inflammation, then alpha-synuclein doesn't get cleared out properly, it builds up. And that buildup is a major driver for forming Lewy bodies.
And those activated microglia we talked about earlier fit in here too?
Oh, they're key to keeping the cycle going. By releasing those inflammatory factors and also damaging molecules, reactive oxygen species, they don't just hurt the neurons directly, they also encourage more alpha-synuclein clumping. So inflammation drives clumping, and clumping drives more inflammation. It feeds itself.
And the genetic links play into this clumping process specifically?
Absolutely. Those genes linked to Parkinson's risk, like SNCA, that's the gene for making alpha-synuclein itself, and LRRK2, GBA, they don't just affect protein handling, they also influence the immune response. So you've got a direct genetic connection between risk, immune function, and the likelihood of forming Lewy bodies.
This whole immune connection seems to be leading us down some, well, unexpected roads.
Yeah.
Including into our gut.
Right. The gut-brain axis idea is becoming huge here. Changes in our gut bacteria, the microbiota especially, that unhealthy imbalance, dysbiosis. It's strongly linked to inflammation, not just in the gut, but systemically throughout the body. It activates those peripheral immune cells.
So trouble in the gut can actually stir up the immune system everywhere else.
That's what the evidence points to. And the theory that's gaining ground is that this body-wide inflammation, maybe starting in the gut, could signal to the brain or even affect it directly. Some researchers are exploring the idea that alpha-synuclein clumping might actually begin in the nerve cells lining the gut, maybe triggered by gut inflammation or bad bacteria, and then potentially it spreads up to the brain, possibly traveling along the vagus nerve, which directly connects the gut and brain.
That's quite a pathway, starting in the gut and moving up.
It is, and it really highlights why people are now seriously looking into whether fixing gut health, maybe through diet, probiotics, that sort of thing, could actually help manage brain inflammation and maybe even affect Parkinson's progression.
Now, this is obviously a really complex picture, and science is moving fast. Are there still, controversies or challenges, things researchers are still wrestling with?
Oh, definitely. For instance, while some genetic studies find overlaps between Parkinson's risk and risk for autoimmune diseases like rheumatoid arthritis, other studies looking at large populations haven't found strong proof that having a classic autoimmune disease makes you significantly more likely to get Parkinson's later. So the immune connection seems more nuanced than just, say, one disease directly causing the other.
And that classic question, the chicken or the egg? Is the inflammation starting the whole process, or is it just the body reacting once the neurons are already in trouble?
Exactly. That's still really tough to disentangle in many studies. Are the immune changes we see a primary trigger, or are they just a consequence of neurons dying and the brain trying to clean up? We definitely need more research to nail down that sequence of events, for sure.
But even with those questions remaining, this whole shift in understanding, seeing the immune system as an active player, that must open up completely new avenues for treatments, right?
Absolutely. It really changes the game for developing therapies. There's a big focus now on immunomodulation, basically treatments designed to tweak or influence the immune system to try and slow or stop the disease.
And what does that look like in practice? What kinds of treatments?
Well, it includes strategies going right after the alpha-synuclein itself. Maybe using specially designed antibodies or even vaccines, trying to help the body clear the misfolded protein or stop it clumping. It also means targeting those specific inflammatory pathways we discussed, like using drugs, inhibitors, to block key players like the NLRP3 inflammasome, trying to calm down those alarm systems.
And given the links to the rest of the body's immune system and the gut.
Yeah, there's a lot of interest in peripheral interventions, trying to influence immune cells or gut bacteria outside the brain, hoping that reducing inflammation body-wide will help the brain too. There are big collaborative efforts now, like the ImmuParkNet project, trying to standardize this research globally, find good biomarkers for this immune activity, and really push forward these immunomodulatory therapies.
This whole deep dive really just highlights how interconnected everything is, doesn't it? The brain, the immune system. Systems we used to think of as pretty separate, and how this new research is constantly reshaping how we understand diseases like Parkinson's.
Yeah, the core takeaway really is quite powerful. The immune system isn't just sitting on the sidelines watching the damage happen. It looks like an active participant, maybe even an accomplice, you could say, or in some situations, maybe even a driver of the disease, especially in forming those Lewy bodies.
And that just changes the whole perspective. It opens up really exciting new ways to think about understanding, maybe even preventing or treating Parkinson's. Looking beyond just the protein handling inside neurons to this much bigger biological picture.
So think about this. If environmental factors, inflammation, maybe things starting in our gut, can influence who gets Parkinson's, and if it's not always about having specific genes, what does that shift really imply? What could it mean for lifestyle choices, for prevention strategies down the line, even for people who don't have a known genetic risk? What questions does this deep dive raise for you?